So the Food and Drug Administration works differently than a lot of people assume it does. A lot of people assume that the Food and Drug Administration has a bunch of labs somewhere and that you send them the drugs or the food and that they do the testing to make sure it's safe. In fact, the Food and Drug Administration acts more like, let's see if I can get this to show up as, resume slideshow. The Food and Drug Administration works more like the Internal Revenue Service. So when you file your taxes with the Internal Revenue Service, they're basically inspect your documents that you submit. And they can conduct an audit. They can go visit you and say, let us see all the original documents that you used to file these taxes. We'd like to, they look at some of the original documents that you send to them. They read your tax form, they see your W-2 forms, they see your forms that show any other sources of income that you had, but they don't ask to see all your proofs of medical expenses, for example. So if they audit you, they're going to come, if you say you have a home office, they're going to come and visit your home and want to see where the home office is. So you better have a home office if the IRS come to call. So the FDA works in much the same way. They have inspectors, and they have people who read all the applications that get filed. But then periodically they send the inspectors out to conduct inspections. So they don't have nearly as many inspectors as there are places making drugs. So they have to be very careful in inspecting the places that have the highest likelihood of having problems. So the FDA, there aren't nearly as many FDA inspectors as probably it would be a good idea to have. So, but people are always, to be an FDA inspector, you really need a lot of background. You have to know a lot about the new drug discovery and introduction. You have to have a background in science. You have to have a background in accounting and bookkeeping. You have to read a lot of lab notebooks and figure out what people are saying. So you know, FDA inspectors are highly trained, well-compensated people. So what is it the FDA is doing then when they're inspecting? They aren't really certifying product liability. The FDA is not saying that all drugs are safe. And FDA approval of drugs does not eliminate the possible liability for dangerous or defective drugs. So if you're a drug company and you bring out that new drug, we talked about this imaginary drug, to regulate male fertility, the male birth control pill. The FDA doesn't promise that this new male birth control pill that you and your new pharmacy company are bringing out will never be defective because of its manufacture, or may not have dangerous side effects. They're simply looking at it, and based on what you presented to them, they concluded that it was safe and effective. So there's a number of reasons that the sellers of the drug could be liable for the harm that that drug does. For example, if in the course of developing the drug, you didn't reveal all the bad side effects of the drug to the FDA, you could be sued and found liable for all the harm done for all the bad side effects. So unlike some areas of the law, the FD and CA Act doesn't preempt state law. So a state can make a higher standard for the safety of the food or the drugs sold within that state. So even though a drug is approved for sale and the drug uses the FDA approved label, the seller could still be liable. So the jury could decide that the seller was negligent because they didn't investigate all the possible side effects. They could also say the company couldn't totally rely on complying with the FD&CA as a shield from the lawsuit. And the company could be liable if there's a link between the violation of the law and the patient's injury. So if you don't send all the information, then the FDA isn't going to make as informed a decision, and you could be liable. So part of what the possible areas of liability for sellers of FDA-approved drugs are based on legal precedent called the learned hand test. So it doesn't mean your hand is educated. There was a judge whose name was Learned Hand. And so what he did was devise an accounting formula for deciding the liability. So B is the burden or cost of taking precautions. P is the probability of loss. And L is the gravity of the loss. So if there's a high probability of loss and it's very grave, then you expect that the burden of taking the precautions should be in there. So if there's a low probability of loss and a low gravity of loss, then the burden of taking the precautions is lower. So this is part of the calculus of figuring out product liability. So again, you're liable if you've concealed adverse information or if you adulterate the product. So if you're selling Viagra and you cut down on the formulation's active ingredients so you can make more money, then that would be a violation of the law. If you have false advertising, if you say Viagra will prevent heart attacks, that would be false advertising. Or if you conceal some of the side effects of the Viagra, that would be false advertising. And if you have inadequate warnings, so the instructions and the warnings on how to use the product safely are inadequate, you're still liable for harm the product does. So this is another diagram looking over the process of bringing a new drug. And so what this does is helps you see how the FDA then interacts with the company while the company is doing this. So the light blue colors on this diagram indicate the time when the FDA is involved in the review. And the little pink triangles indicate what kind of documents you're submitting to the FDA and what kind of documents you're getting back from the FDA. So when the IND for the drug is submitted, that's when you're beginning to enter the clinical studies. And when the new drug forms are submitted, that's another time when the FDA is going to review your new drug. And you're going to be submitting those after your phase 3 trial. And here you see your phase 3 trial. There's several yellow diamonds when you could be meeting with your FDA advisors. So if you're having questions about what kind of studies to devise and what kind of data analysis the FDA wants to see from those studies, you could speed up your new drug interaction by scheduling a meeting with your FDA advisors, so they would give you feedback on what they're looking to see at those particular times. And there's advisory committee meetings where you sit down with the FDA and they give you questions. So you can be sent back to do more work, essentially. Or you could be sent back to rewrite your new drug application. So those interactions with the FDA then, the more experienced you are, the better you are at figuring out what the FDA wants to see when you submit a new drug application. So that's one of the reasons they like to get experienced people on board in a pharmaceutical company, because those people are better at figuring out what you need to be sending into them. So what these clinical trials, I'm giving you another look at it. Here you can see what kind of population you're working with here. So in the preclinical testing, which can take a lot of time, you're spending a lot more time sometimes devising the test and making sure that you've got statistically good numbers on the test. So those are the preclinical testing. You're doing laboratory tests and animal studies, and you're really trying to assess safety and biological activity. And you could be looking at 5,000 different compounds in preclinical testing. And of those 5,000 compounds testing, only 5 might enter the clinical testing trials. So out of those targets, then, you're working with small groups. So in some phase 1 studies, you're only doing 20 to 80 healthy volunteers. In some cases, for rarer diseases or for cancer, you might only be doing 10 in an initial phase 1 trial and then expanding it. In a phase 2 trial, where you're looking at effectiveness and side effects, you're maybe only looking at 100 to 300 patient volunteers. And those people may not be healthy people. Those are people with the illness you're treating. So the healthy volunteers, in the case of a chemotherapy drug, you may still be dealing with people who are individuals with the cancer. So even though these are typically healthy volunteers for some drugs, for other drugs they're patient populations. So then phase 3, you're doing big trials, 1,000 to 3,000 volunteers. And now you're monitoring for adverse reactions from long-term use, and you're verifying the effectiveness of the dose that you've selected and the treatment regime that you've selected. So out of that, then you're going to file the new drug application. And so that new drug application, then, only one of those may make it through those initial clinical trials. So over time, then, on this schedule, it could take as long as 12 years to bring the drug out. And then, in phase 4, the FDA may require additional post-marketing testing. So the basic disciplines of drug development then are the chemistry, manufacturing, and controls, the non-clinical aspects, and the clinical. So here, you're making the drug, you're coming up with new compounds, you're figuring out how to manufacture the drug to an appropriate level of purity, and how to determine whether or not the test is effective or not. So those initial parts are almost preclinical. And then you get non-clinical testing on rats, mice, cell cultures, and then clinical testing, you're dealing with humans. So the chemistry, manufacturing, and controls part of drug development, you could be just synthesizing a bunch of compounds and then screening them all. You could be, just by luck, have made an observation and noticed that something could be a really good drug. That serendipity is what the people who put the Rogaine observed. They were looking at a different trial for a high blood pressure medication, and they found the side effect of growing hair. Folk medicine can point us towards potential drugs that could be obtained from those folk medicines. So, for example, looking for drugs in Ayurvedic remedies, looking for drugs in turmeric, looking for drugs that could be obtained from Chinese, Asian medicine. So, as part of that, after you can also do rational drug design, where you understand the mode of action of a particular receptor, for example, and you design a drug to block the receptor. If there's a receptor that the HIV virus is relying on to transport it into the cell, and you come up with a drug that blocks that transport by rational drug design, that would be a good drug. So besides the discovery phase, there's the synthesis, purification, and scale-up of the drug manufacturing. There's the analytical stage where you figure out what is exactly the structure of the chemical that you've made. What is its activity? What kind of, how do you have to package that? Do you want to put it in a tablet? Do you want to put it in an injection? Can you give it to people as a liquid? Does it have to be intravenous? How pure is it? How stable is it? And then pharmaceutically, you're looking at what's the dosage form. Do you give the drug once a day? Do you give the drug twice a day? How is it administered? What is the packaging? And most importantly, what is the labeling to preserve the qualities of the drug that you've gone to such efforts to develop? So the FDA is relying then on high quality records. So since the FDA isn't testing all these drugs for effectiveness, they're just auditing your records, they need quality records. So the record keeping process is what's known as good laboratory practice, GLP, good clinical practice, GCP, and good manufacturing practice, or GMP. And so this record keeping process is very carefully defined by the FDA. So for example, in GLP or GCP or GMP, when you write something down, you don't write it in pencil, you write it in ink. If you write it in ink and you change something, you made a mistake in what you wrote down, you have to draw a line through it and not obliterate it with whiteout. And you have to initial and date that you've changed something so that they can tell who made the change. So those kinds of quality changes in the records are very critical. So good laboratory practice is the fundamental underpinning of a lot of this, and it ensures the quality and integrity of the data by establishing basic standards for the conduct and reporting of non-clinical safety studies. And then there's guidelines related to studies in animal models or other research as well in GLP. So the GMPs are sometimes referred to as the CGMPs, or current good manufacturing practice. So those are guidelines related to manufacturing practices and specifications. And so the focus on CGMPs is often on impurities. How pure is the final product? And it's necessary to ensure the quality of the drug product and the drug substance. So if your finished dosage form isn't equally pure every time you make it, you're going to have problems. So drug development, then the non-clinical testing, the objective is to develop a pharmacological profile and to determine the acute toxicity, how much of that drug will cause a toxic effect. And you have to use at least 2 species. So maybe a rodent species and a dog species, for example. And to assess toxicity in studies that could be from 2 weeks to several months. So it doesn't give you long-range toxicity, but acute toxicity as well as gradual toxicity. So when you're talking about toxicity, you're trying to figure out something called the therapeutic index. So the therapeutic index compares the amount of therapeutic agent that causes the therapeutic effect to the amount that causes the toxic effect. So the ratio of the dose required to produce the toxic effect is divided by the therapeutic dose. A commonly used measure is the lethal dose of the drug for 50% of the population, divided by the effective dose for 50% of the population. So the LD50 is divided by the ED50. So as a goal then, you want a therapeutic index where it's a higher index. So if you need a lot of material to get a lethal effect compared to an effective dose, that's a relatively safe drug. If your LD50 and your ED50 are too close together, then that's a relatively risky drug for therapeutic index. So the biologics then are any virus, therapeutic serum, toxic, antitoxin, vaccine, blood or blood component or derivative of blood, an allergenic product or analogous product, or arsphenamine or its derivatives. And arsphenamine is an organoarsenic molecule used in treating syphilis. And so when you're looking at biologics, they're included. And the application to the prevention, treatment, or cure of disease or injuries of man. So if you're making a vaccination, if you're giving a blood clotting protein, if you're giving an antitoxin to counteract a toxin like an antivenom from a snake bite, that's a biologic. So in 1902, there was a Virus Serum and Antitoxin Act passed that was part of the CBER, the Center for Biologics Evaluation and Research. And so that means that you have to be licensed as an establishment. So if you're the Red Cross, one of the things they do with all the blood they collect is they purify components of it. So they make plasma, for example. So every Red Cross blood donation area has an establishment license, and every product made from the blood has a product license. Every product made is labeled, all the facilities are inspected, they can have their license suspended or revoked, and there's penalties for violating the safe following the Virus Serum and Antitoxin Act. So, as I've said, they're also part of analyzing somatic cell and gene therapy. They're also part of transplant medicine and tissues. They're also regulated when biological products are used in vitro diagnostics. When there's extracts that could be allergenic, monoclonal antibodies are regulated by CBER, and biotech-derived therapeutics. And when you're getting a transplant from some other animal, for example, some bone transplants, or some valves are transplants from pigs. The cartilage in heart valves can come from pig hearts, which are similar in size to human hearts. So that would be a xenotransplant. So medical devices are also regulated by the FDA. So the medical devices could be something as simple as the device for reading your blood sugar, it could be your oxygen, it could be your blood pressure cuff, or it could be something implanted into you like a stent. So before marketing a medical device, the manufacturer has to submit a pre-market notification called a 510K, or pre-market approval, PMA, application to the FDA. So the 510K is a marketing application submitted to demonstrate the medical device is as safe or effective or substantially equivalent to some other legally marketed device. So if you're submitting a blood pressure cuff, you have to show that yours is as effective or as safe as the blood pressure cuffs currently on the market. And so most devices are getting a 510 process. The pre-market approval is the process for the class 3 devices. So those are the more invasive devices. And those pre-market approvals need clinical studies. And they're subject to investigational device exemption regulations as well. So if you're investigating if it works, you can get permits to try the device. So I will leave you here, and we will talk next about how you screen and identify compounds and identify which areas your drug company should focus on, and how you identify which are your new candidates for drugs.